Hello, hello.
Hello.
So we're going to start in uh, maybe 2 to 3 minutes. I was hoping more mm biologists will come in as well. Yes. I think our token biologist today is Devashish. So we are going to bother him. Devashish, where are your colleagues? Maybe about so. I'll just wait for 2 minutes, Jan. Okay. नहीं उसको ना हाइड हाइड कर दो ना नीचे हाइड हाँ ठीक ओके ओके अगर आ गया तो ठीक 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 So there's a faculty called Alak. I don't know. Oh, he was for some time with Anfinson. Not Anfinson. No, but the biophysics. Leventhal. No, he was with Leventhal. He was with Leventhal. No, Barry? Barry Honig. No, but he. Then he. Yeah. Okay. Barry Honig, of course. हमारे बंदे किधर है? Okay. Oh, there's a token biology student in the audience. Oh, no. There are a couple more. So thank you, guys. Maybe you can tell your group members to also attend. Yeah. Okay, you already know. Thank you. Okay, I'm going to start now. So um, there's been a bit of a hiatus from the colloquium, well, 1 week at least. There were chemistry interviews going on, and I think we are going to back again, have the next week will be physics interviews, so we'll not have the colloquium again. But sandwiched between these two absent weeks, we have a wonderful speaker today, Professor Jayant Udgaonkar, who is currently the director of IISER Pune, but as far as I remember, has ever been associated with the TIFR system and NCBS, the National Center of Biological Sciences. And I, of course, would like to say a few more things before Shudipto actually formally introduces him. But for all who are joining the colloquium series for the first time, let me remind you that this idea of the NSF Wednesday colloquium was first conceptualized by Professor Homi Bhabha, our founding director, who actually found it very nice to have all the different disciplines of sciences get together one day of the week and listen to an eminent expert in any of the natural sciences, areas of natural sciences. And that really has, if the younger generation is please listening, has trained lots of very inquisitive minds that actually don't think about science in their own category, but actually are looking beyond, not just from the molecule, but the stars. So you need to look beyond. And this series is actually a platform for that. So, so those who are coming in, thank you for joining and believing in this. The last thing before I hand over to my department chair, Professor Sudipto Maiti. I would like to say just a personal story, and that is when I was actually interested in biophysics, and I'm still interested, but I just don't do work there. But in my PhD days, Jayant's name was formally introduced because of the protein folding course that I took from one of my faculties at Princeton, Janet Carey, who talked about the molten globule and the work that Jayant and his PhD advisor, Robert Baldwin, had done in Stanford for some time. And this is something that sort of had put a mark on my mind that if I were to go back in India, and I would like to meet this person. And that happened soon enough when I was in TIFR, when I joined. And it has been an immense pleasure to know Jayant over the years. Not because he has been doing some great science, but there are many people who do great science, but a nice person to talk to. That is rare. So I would like to let the other parts of the introduction to Sudipto.
Thank you, Jyotishman. It's difficult to come into Jyotishman's steps, but we are really glad to have Jayant here. Jayant has, so it's really, I shouldn't be introducing Jayant to TIFR. It should be Jayant who should be introducing TIFR to you, because he is a second generation TIFR faculty. His father, Professor B.M. Udgaonkar, was one of those stalwarts of the theory physics department. And this is, of course, they started before this campus housing was built. So he didn't actually grow up in the campus, unlike, let's say, Shubha or somebody. But he has obviously been very, very familiar with the campus. You know that Professor B.M. Udgaonkar also was instrumental in setting up the Homi Bhabha Center for Science Education. So anyway, so let me get back to Jayant. So Jayant grew up in Bombay, went to St. Xavier's. I can see another Xavier right over there behind the mask, Vidita. So he went to St. Xavier's for his bachelor's, went to IIT Madras for his master's degree, and then he went to Cornell, okay, where he worked with George Hess on a very biological problem. It's a biochemistry, which is looking at acetylcholine and acetylcholine receptors on the cell surface. I remember Jayant later on. I didn't know Jayant at that time. It's not yet at Cornell. I would come into Cornell maybe 7-8 years after you. Jayant. So talking about how to release caged neurotransmitters. And since I had some interest later in neurotransmitters, we started talking about that before we started talking about protein folding. So later when I met him. Anyway, so he made a name for himself there, and then he moved to the area of protein folding, where he really is, he's our protein folding man in India. So he went to and joined Bob Baldwin in Stanford for his postdoc. And again, just like Jyotishman heard about him before he actually met him, I read his paper, Udgaonkar and Baldwin, about looking at the protein folding intermediates and the technique of hydrogen exchange NMR. And this was actually something that we didn't like very much, because I was a graduate student there, and two of my own professors were also competing in that technique, Heinrich Roeder and Walter Englander. And I think their thing came up first, or about the same issue. And obviously they have been pioneers. So Jayant has been a pioneer ever since. Then he came back and joined NCBS of TIFR, which didn't have a physical existent yet. End of 80s, early 90s, and 91, he said, right? So he had a lab right in DBS here as part of NCBS. Then he moved to, NCBS moved to this building in IISc campus. Then they had their own building where he had been since then, and really has been a trailblazer in the area of protein folding. He showed how, so in one of his initial Nature papers, he showed how the hydrophobic collapse, he worked with a lot with a protein called bar star. Hydrophobic collapse actually happens before you get secondary structure. It was talked about, but not very much proven till then. And then he did, many very excellent work, some together with our department colleague G. Krishnamurthy, and also a theoretical physicist. He actually talked about um, friction in protein folding. This was like 2012, 13 kind of thing, right? Yeah? I had a PNS paper, which was again a trailblazer in that area. Then he got into an area which was close to my heart, which is protein aggregation. And protein aggregation, he has again done excellent, astounding stuff. So till 2016, he was physically at NCBS. Then 16 or 17, he took up the job of being the director of IISER Pune, where he still is as the director. So obviously, he has won all the awards, Bhatnagar, etc. He is member of all the academies in India, but those are smaller things. It's what he has done for protein folding in India that really takes. So here, waiting to hear from you.
So thank you, Jyotishman and Sujito, for inviting me here. I last gave a colloquium here 33 years ago, one or 2 months after joining NCBS. And so it's good that towards the end of my tenure at TIFR, I'm getting a chance again to talk about my work. I'll be talking about my perspective on protein folding. And so it's of course got my biases in it. I'll try to give you a general idea of the field, but again, it'll be colored by what I think is right and perhaps what I think is not right. And let me begin. So when we talk about protein, folding, why is it not changing? It's changed here. Not moving.
Oh, this is the one. Sorry, this is switched computers.
Oh.
This switched computers. We are not using this. We switched computers.
Okay.
Yeah, I'm sorry.
That's okay. So long as I know which one. So this way I'll be standing right in front of the slide. Can you move it this way a bit?
That can work as slide changer.
Okay. I'm just clumsy with this, so it's just okay. So let me. So, a protein is a polymer. So it's a chain which is synthesized on a machine in the cell, and the information for how it is coded for is in the RNA, which comes from the DNA. So it's a necklace of beads of 20 different colors, and because it has 20 different chemical moieties, amino acid residues in it, which is, so you can, so this picture shows it coming off here, or you can see it coming out through a tunnel in the ribosome out into the exterior in the cell. Unlike typical polymers, which chemists are familiar with, the protein chain adopts a very well-defined structure where the positions of atoms are precise to fractions of an angstrom. Okay? And the information for the structure is coded for in the amino acid sequence, so the sequence of colored beads. And as I said, unlike necklaces of beads, this necklace folds into a functional structure. Okay, so what happens is this necklace folds into a structure which gives function to this protein chain. Now, you might ask why this is important. It's important because the whole idea of having this structure is to provide a scaffold for the active part of the protein. So here are two proteins which you might have heard of. This is a protein in your gut which digests your food. This is a protein with similar function, which is produced by bacteria, and those of you who use Tide detergent will be using subtilized this is present in that detergent. Apparently removes protein stains, if you believe that. But what you can see is that the positions of the three active, this active part of the protein, the orientation of the three amino acids is essentially exactly the same in the two different, completely different scaffolds. So the purpose of the scaffold is just to bring the active part of the protein together, the atoms in proximity, for there to be function. So as I said, the protein folds when it comes off the ribosome, but proteins are folding and unfolding all the time in your cell. And that's because they have very marginal stability. So a protein during its lifetime in the cell may fold, unfold millions of times all the time. So it's not just happening at the time of synthesis. And it's also happening all the time in a variety of functions. So you perhaps might remember from school about how muscles contract. You have two proteins, actin and myosin, which move with respect to each other. And if you have this type of motion, you would want a rubber band to make sure that you don't get out of register too much. And the molecular bungee, which performs the function of the rubber band, is a protein called titin. Some of you would be familiar with it because Koti here has worked a lot with it. And the way the rubber band works is to make it long, certain protein domains unfold, so it becomes long. And when it has to become short, the protein modules refold. So that's how your rubber band becomes long and short during the functioning of every time any muscle of yours contracts. So this is a problem which has been there for several decades. And the question really is, it's been a difficult problem to work on, mainly because many of the processes involved are very fast. And so experimentally, it becomes very difficult to characterize them. And theoretically, computational methods have yet not been able to determine mechanisms of how structure is achieved. There's been a lot of progress, especially through AI-based methods. So many of you would have heard about AlphaFold, which can now predict protein structures very, very well. And so that has addressed one facet of the protein folding problem. How do you predict structures from knowing the sequence of amino acids? But it hasn't helped much in the question I'm interested in. And that question is, what is the mechanism or the pathway of protein folding? Mainly because there is lots of human-derived data on proteins for which AlphaFold can use to predict structure, but there isn't enough understanding or data on how proteins fold. And so, AI-based methods have not had much to learn from. So experimentally, what one thinks about when one talks about the protein chain folding is what happens first, what happens next, etc. And there have been phenomenological models to assist experimentalists in their thinking. And so the question, for example, as I said, I'm clumsy with this. So it does, for example, what are called secondary structures, helices and sheets form early, or does the protein chain collapse, as many polymers collapse and then form structure, or do nucleation events happen which then propagate and then structure in the protein? So these are ways of thinking about the process. Now it's possible, for example, that all three processes happen for the same protein under different conditions. And those will be questions which I will be referring to as the talk goes along. So if you look at what are called energy landscapes, so you have an unfolded chain going to a more stable native state. What is the energy surface which the molecule traverses to reach this state? So you can talk about a transition ensemble or an ensemble of microstates which have to be gone through to reach the native state. So this is a one-dimensional plot. In a two-dimensional plot with two coordinates, for example, You could have a coordinate of size and a coordinate of number of native contacts, the contacts between parts of the protein. You could have one trajectory which is going through all these states coming into the native state. And you could have another trajectory which is going as shown out here. Now, the question, for example, is structurally speaking, how similar is this trajectory to this trajectory? In other words, does, for example, if the protein has two helices, does helix 1 form first here and helix 2 second, and here helix 2 forms first and helix 1 forms last? So these are the type of questions which we have been interested in for quite some time, as have been most of the field of protein folding. Now, as you can see, the transition state ensemble of microstates is surrounding your native state in this depiction. And you have to go through this. It's not one unique state, but many different states, microstates, which have to be traversed to reach the native state. So what's the role of this, and how are these conformations different from all these other blue conformations shown out here? So there's been a lot of advance from theory, from statistical mechanics on how proteins fold. And I just want to give you, since there are physicists in the audience, a small little picture or tutorial on energy landscape. So you could consider, for example, a toy model where you have a necklace of 6 beads, and you have hydrophobic beads, and you have polar beads, as shown out here, orange and blue. And the structure gets stabilized by interactions only between the hydrophobic beads. And you could have either microstates with 1 interaction, and you could have microstates with 2 interactions, and these are all microstates without any interaction. So this is more stable than this macrostate. This is the most stable. And so you have 28 possible microstates here, 7 out here, 1 out here. So your partition function is straightforward. And then you can make a plot of the probability of having U or the folded or this intermediate state as a function of temperature. And you will see, for example, sigmoidal transitions as shown out here. Now, if you now make a plot of conformational count versus the number of contacts, you have this. So you're going from 28 to 7 to 1 in this. If you flip this by 90 degrees, you will get this. And this is your what's called the folding funnel. Okay. You have many microstates out here, fewer and fewer as you get down to the native state. So here in this case, your microstate with only 2 contacts is your native state. So this is the folding funnel, and this picture is taken from this book by Kendall and colleagues. So this type of idea about folding free energy surfaces has been there now for 20 to 30 years. And as we saw in the previous slide, the x-axis is configurational entropy, the number of possible states, and the y-axis, the ordinate, is effective energy. And so you end up with it's like a free energy surface because there are many states out here and fewer out here. So entropy changes as you go from top to bottom, and hence free energy changes. So, and this is a more colorful depiction of the same. Now, the thing is, theory in protein folding, statistical mechanics theory, has advanced to a stage where it's claimed that it describes protein folding, and there's nothing perhaps much more new to learn. The problem is, well, it's true that the theory can accommodate all observations, but it cannot predict what will happen in any one system. So how useful is a theory which can only accommodate all observations, but not be able to predict and describe what happens for any particular protein, or even end up with general ideas of how, why does this, okay?
It's not.
Okay, so as I said, what happens in actual proteins is unknown, and both computationally and experimentally, it's impossible to predict or to determine the folding trajectories of any one given protein. So, so the question is how do you reach the native state from this very large number of conformations which are present in the unfolded state. So if you have, every molecule in the unfolded state has a different conformation. The native state has essentially one conformation. And the analogy can be made to evolution. So we all have very complex structures, for example, our eye. And one can ask, we probably all ask ourselves, how do we arrive at, how were these structures arrived at during evolution? And Richard Dawkins, in one of his books, made an analogy to a monkey trying to type a sentence from Hamlet on a typewriter or on a computer keyboard. And you might think typing a sentence like me thinks it is like a weasel would be impossible for the monkey to type and would take forever. But if something very simple was done, which is that if a correct letter is typed at the right position, that is not allowed to change, then within 3,000 typing events, the correct sentence is arrived at. So you can see the pattern of green developing with a number of attempts. You put a second monkey in front of the same typewriter, you get another pattern of green. The second, what's happened to the bottom of the slide? So again, in roughly 3,000 attempts, you have come with the correct sentence, but the pattern of green is different, which means that the formation or structure during folding is different, or the structure forming the sentences is different. Okay, so there was a very well-known paper from Zwanzig and Szabo and Bakshi, three of the most eminent physical chemists alive today. Maybe it's one. And this picture is from a well-known biochemistry textbook from the 90s. So what this tells you is that if you have a bias search, like what the monkey is doing, you can have folding happening in biologically relevant time scales. So there was a well-known paper which was published in the 1980s by Harrison and Durbin, which likened folding to the assembly of a jigsaw puzzle, that you have a completed jigsaw puzzle, that's the native state. You all know you can assemble a jigsaw puzzle in many different ways. Is that how proteins fold? That for different molecules, structure forms in a different way. So these are ideas which have been there in the field, but they've been very difficult to resolve until very recently. And I'll be talking about some of our work later, which tried to address this. So the questions which have motivated us and also many others in the field are really questions related to multiple ways of achieving structure. How different are they, and how do you show how they are different? We have been very interested in chain collapse, what happens very early during folding, but I won't be talking about this at all. We've been interested in the roles of partially folded structures on the pathway, but again, I won't talk about this. What I will talk about are these two questions as well as the first question. What is the nature of the barrier which slows down folding? Okay. So you might think on an energy surface such as what I showed you, the funnel type surface, you won't need an activation barrier, but invariably you do. And what the origin of that barrier is something which is still not properly understood, whether it's completely entropic in origin, because remember, you're going from a state with many, many microstates into a state which has got just a few microstates, or are there specific interactions which have to form, which constitute, and the formation of which constitutes the barrier to folding. So we've been looking at folding over very different timescales with many different probes, and that's perhaps how our lab work has differentiated from many other, nearly all other labs. And we've been able to go down to very fast measurements. So we built, for example, set up to do microsecond measurements of folding, which you cannot do on commercial instruments. And if you use many probes and you go over a fast time scale, you can obviously identify many different partially folded structures. I'm not going to be talking about this, as I said. I just want to illustrate an important point, that how does one know that one is able to, even if you go to faster and faster time scales, use more and more methods, how is one sure that one is capturing everything on that time scale? And that is a problem which is difficult to answer. So for example, if you have this type of a reaction coordinate diagram, this is what chemists, small molecule chemists typically use. The coordinate, for example, could be number of native contacts in the protein. If you have this type of scenario, then this fellow is more stable than this and more stable than this. So this will accumulate during the reaction and you'll be able to detect it. But suppose your folding conditions are such that this fellow is higher in energy, then there's no way you will detect it and you will not know it exists at all. And so you would not know what the mechanism of folding is. If you did the experiment in this condition, you would say, there's a early structured intermediate. If you did it in this condition, you would say there's no such structured intermediate early during folding. So these are questions which have been very difficult to answer. And unless you not only have good time scales, many different probes, and also look at folding under many different conditions, you're never going to be able to answer questions such as this. So coming back to the question of barriers, the question or the ideas in the field have evolved. And in the 90s and 2000, there was a lot of emphasis on what was called two-state folding. It started being believed based on, I think it was an erroneous belief, that folding could be two-state, that you go magically from the unfolded to the native state in one step. And so the idea is that if you typically unfold a protein by putting it in urea or some other chemical denaturing or changing the pH, and you go back to native conditions, you will get a transition such as this. This is where it's folded, this is where it's unfolded. And the idea is at the midpoint of the transition, you have equal numbers of the folded and unfolded. So you're really two-state right throughout. And your properties of the system are described by just your native and your unfolded. Now, this is something which was very attractive to experimentalists because it made things very simple but incorrect. Okay. And so, for example, what might appear two-state is not really two-state because there are intermediates which are populated to too low an extent to be detected. So, for example, if you have a real two-state system would be like this, but if you have a high energy intermediate, there's no way kinetically you can distinguish this from this or this from this. You can distinguish it when this fellow is more stable than this, but otherwise in these type of scenarios, you can't distinguish from a two-state system. And it's possible, for example, there are many, many forms up the barrier and down the barrier. And you could, for example, have a continuum of intermediates. And this case would still appear like this in experimental studies. So what exactly is happening?
What exactly is happening? Okay, I thought it was a thoughtful question.
No. Well, I didn't mean. So we talk about a barrier. Now the question is there a barrier? So barriers are meant to slow down folding. Okay. And a free energy barrier would appear when there's a mismatch between entropy and enthalpy changes during. Now it's very difficult to believe that during protein folding, the mismatch would occur only at one point on the reaction coordinate. But that's what this type of picture implies, that there's only one little point on the reaction coordinate where you have a mismatch. It's more likely, given the complexity of the reaction, that you have many mismatches and hence many small free energy barriers. In this type of scenario, folding would be slowed down by having to go through all these little barriers. So just like a marble coming down an inclined plane would be slowed down if there are a lot of indentations on the inclined plane. Okay. And so is this the correct picture or is this the correct picture? Is part of the reaction like this and part of the reaction like this? Can it be that the entire reaction is like this? So these are questions which have been there in the field and which we too have been interested in. And
Jen, a quick question.
Yeah.
You had that illustration of the reaction coordinate of a complex surface the free energy surface. And you mentioned that, of course, depending on the conditions, stabilizing, marginally stabilizing versus strongly stabilizing conditions, you can alter the intermediate energies.
Yeah.
But that has been true for even for the folded and unfolded states.
Yes.
The folded and unfolded states energies can also be tuned. The urea lets you see the unfolded state.
Yeah.
So is that a thought-provoking idea to understand the free energy surface? It's always going to be like that, right?
Yeah,
You
but
don't learn much with
sometimes
that.
simple ideas are ignored.
Okay.
So it's not a question of, so one, the stability of native and the unfolded depends on the conditions. The structure in general is not supposed to, okay? An intermediate which forms on the pathway, its population will depend on the conditions, but people, when especially experimentalists or even computational people, will be saying that, oh, there is a pathway with this as a kinetic milestone. They don't take into consideration, oh, it's probably buried in the recesses of the head, this thing which you say is obvious, and it's ignored. Hmm? And until someone states it explicitly. So the question which I just was referring to is that, is folding happening in an all or none process? So if there's a barrier between U and N, where you have one population with one energy, another population with another energy, and this goes up and this goes down, or the other way around. So this is either here or here, all or none. Or is it happening gradually, where you have a monomodal distribution shifting gradually on the energy scale? Okay. So this question is an important question, and it's been very difficult to address experimentally. And those physicists who are interested, perhaps might be interested in physics, would look at this book. It's worth looking at. Yeah.
Yeah, with respect to the denaturation experiments, right? You're using urea.
Yeah.
Right. Instead of urea, I mean, if you use something like pressure or temperature, for example, do you get better resolution in terms of looking at, like, states en route to the unfolded state, for example?
Well, the structural resolution is going to depend on the probe you use to monitor structure. So you could typically use the same probes for all three methods of triggering the reaction. The timescales of the reactions might be different. So if you're doing a urea dilution experiment, you're limited by how fast you can mix and dilute out the urea. Whereas if you do a pressure jump, pressure jumps can happen in nanoseconds.
Right.
So you can look at the reaction from nanoseconds onwards.
Right. That's what I mean.
Yeah. So the temporal resolution would be better with some of these methods if you do temperature jump or pressure jump. But the structural resolution will depend on what probe you use to monitor.
Okay.
Yeah. So we have been interested in this question of what is the best way to describe the energy surface. And as Sudipta mentioned, we had a long association and collaboration with G. Krishnamurthy here at TIFR. And we started questioning this two-state model nearly 25, 30 years ago, with this paper where we looked at anisotropy during a chemical denaturant-induced unfolding. And we ended up by talking about a gradual transition. This was somewhat descriptive because we had no real way of describing what exactly happened. So we then, along with GK and his students and my students, we started using FRET to look at the process. So I presume most of you are familiar with FRET. What you have are two fluorophores, or a FRET donor and a FRET acceptor, and energy transfer happens between the two through dipole-dipole coupling. So there's an R⁶ dependence, and FRET can be used as a molecular ruler, where the efficiency of transfer depends on the distance separating the two. So you can use FRET to monitor how distances and then structure varies during the folding, unfolding reaction. So we did this, and we did this with this initial work with GK using FRET. And what we do is measure fluorescence lifetimes, which become short due to FRET. And what we could tell is that the native state, remember I showed you an unfolding transition where it looks flat out here when you measure it by probes such as fluorescence or CD, etc. But if you did it by FRET, what is happening in that region is that the molecule is continuously expanding. Okay, so structure is changing, and then it goes through some free energy barrier into the unfolded form, where again it expands. As you can see, the lifetime is increasing both here and here in both the native and unfolded forms. And that's what's happening. So you have expansion in the native, a first order transition to U, and then expansion in the unfolded. So this was the first real demonstration.
Jayant, I have a question. So this is actually separate steady state measurements at different unfolded quantitative concentrations. This is not really what is happening in time.
Yeah, so I'll come to that.
Okay.
Okay. So these are you always do the easy experiment first. So it's always best to establish endpoints and before doing any kinetic experiment. And so this was the first real indication of continuous change during folding, unfolding. And subsequently, there have been a whole series of papers using NMR where you look at chemical shifts of individual resonances, and you find that they don't overlap with each other. And this is with the prion protein, one of the proteins I work with. But if you take an average of all of this and compare it to a fluorescence measured transition, you'll find that the overlap is quite okay. But the individual traces for the individual atoms is quite different, as you can see out here. And Hosur at IIFR has done similar experiments with a variety of different proteins. So we started working with this protein called monellin. It's famous as probably the sweetest substance known. It's about 100,000 times sweeter than sugar. But we work with two variants of it. The natural variant is a two-chain protein. There are two polypeptide chains held together by the structure. And we created an artificial variant by connecting up the two chains at the gene level. And so this is the experiment with temporal resolution. And so what we did here was look at 4 different distances in the protein as the protein unfolds with time. And this again work with G. Krishnamurthy experimentally. And what we found is quite very different from what we expected. So for example, at one of the early time points, the unimodal distribution for the native state becomes bimodal, which means there are 2 populations of molecules. And then the 2 populations evolve with time. This fellow becomes this, finally. Okay, all of these transit into this. So if you measure this part of the molecule or this part of the molecule, that's what you see. But if you measure this distance, you only see a unimodal distribution changing with time. So what this means is that this distance during unfolding is continuously expanding as you go, as the molecule unfolds. And this 4th distance, which he looked at, has components of both these 2 behaviors. There's both one population going into the other, as well as a shift of each of the populations on the distance scale. From FRET, you can get the distances out. And we didn't know how to analyze this data, so we went to Deepak Dhar, and Deepak Dhar first told us that this is obvious. Then he said, but people, we don't know how to, we have some qualitative idea what's happening. We want to treat this quantitatively. He said, it took more than a half an hour or 45 minutes to convince him that it was a problem worth his time to look at. And then he said things like, there are some things which are so obvious that you don't need to show any, you don't need experiments to demonstrate that. I don't know how many theory people believe that. But he said that. But anyway, he finally, with his help, Santosh, my student, we analyzed the data based on the Rouse model of polymer physics. And I had a lot of problem in, this time scale is also over the seconds. I had a lot of problems about a protein molecule swelling over so slowly over this time scale. And Deepak said, well, you can buy these polymers on the roadside where they swell very, very slowly. So I didn't know how to argue with that. But anyway, the idea here is that you go first to two populations, each of them expands, and then one population transits to the other over some barrier during the unfolding process. So this method is good, the FRET method, but it's somewhat cumbersome because for every distance, every part of the segment of the molecule, you need a new FRET pair, etc. And the experiments are not easy to do. So we went to do hydrogen exchange experiments, and I'll spend, yeah.
Sorry, from the previous slide, would it be harder to do this experiment for a fast folder?
For a
fast folder?
This is for a slow unfolder.
Yeah, or slow, a fast unfolder. Yeah.
Oh, we'll come to we did the experiment for unfolding first because unfolding is invariably slower than folding.
Okay.
And
yeah,
it took us another 9 years to do the folding experiment.
Okay.
Because technically it was more difficult.
And each of these cases, this is urea denaturation?
We did guanidine denaturation. Guanidine is like urea.
Yes. Sure.
So you unfold it in guanidine, dilute out the... So we decided to use this method called hydrogen exchange. So this is a pretty simple method, but what you do is, the peptide bonds in proteins, the NH-CO bonds, the NH can exchange with the deuterium or hydrogen in your solvent, in your water. So if you take your protein and dump it into D2O, then all the NHs which are accessible will become ND, okay? And so for example, you can see that this protein has got all Ds at the NH sites. So if you have 100 residue protein, there are 99 of those sites, and all 99 will not have deuterium. But let's say you start off with everything deuterated, you can do that. You dump it in water, some of the deuterium remains, some becomes replaced by protium, hydrogen, okay? And the protected parts, protected by structure parts will retain the deuterium, other parts will exchange, and finally everything will exchange because the protein is not a static object, it is fluctuating and everything will exchange. So what happens when you have deuterium and hydrogen? The mass of this fellow is different from this, okay, because I'm really clumsy. Okay. So, and you can easily detect these shifts of a few Dalton by doing mass spectrometry. Okay. So as H replaces D, the mass becomes smaller and smaller. Okay. And you can look at the kinetics of deuterium getting out of the protein and being replaced by water. And there are good, so this method is very good because if you have a structure which is completely unfolded, everything will get hydrogen in it. If something is native and has protective structure, everything protected will have D, everything else will have H. And if you have an intermediate structure, the protected part will have D, and this will have hydrogen. So the masses of these three species will be different, and you can differentiate between the three when they're present together. So I don't really want to describe this in detail, but for exchange to happen, you have to have opening out of the structure, and then the D can be replaced by H, or H can be replaced by D. That's what this is telling you. And you can do the experiments under conditions where you're limited by how fast this opens. You don't want to do it under conditions where this is very fast compared to the intrinsic exchange rate, which is replacement of the D with the H. Okay, the second step out here. So I'll skip this. So what we did was, we took this protein, it's the simplest experiment you can do. We started it deuterated and we dumped it in water. And what we found is that the mass just shifted in a completely gradual, continuous manner. It's like, this is my protein, it's got some protective structure, it's losing the protective structure like this, like this, like this, like this, like this. Okay? Step by step, every part of the protein is unfolding and losing its structure. Okay? Remember, we are doing this under conditions where what is limiting is the rate constant of opening. Now, we did this in zero denation. Finally, over 36 days, everything exchanges. Everything exchanges when the entire protein opens out. So it's like measuring unfolding in the absence of any denaturant, okay, in water. It's that slow. But we added a bit of guanidine, the denaturant everybody uses, and now it's continuous for a short time, for 3 minutes, and then it becomes bimodal. So it's like going like this, like this, like this, and then the rest of it opens up. Okay? So it's cooperative, the latter part. Here it's not cooperative at all. Okay, by cooperative, I mean things happening all together. So how does one explain this? And fortunately, I teach every year. And in part of my course in physical biochemistry, I teach about Marcus's electron transfer. And he has this review where he has these type of pictures. And protein folding and electron transfer don't have much in common, but he had these figures. So that's what it says, inspired by this paper. And the idea is very simple. So if you have a barrier, what does it mean? You have a native well and unfolded well. And to go from one to the other, you have to climb the barrier and go across. And the resultant free energy surface is the one computed in gray. Okay? But suppose you stabilize one or destabilize the other. Suppose you stabilize the native state, you're pulling it down. Now there is no barrier left. Okay? If you destabilize you, again, you will have no barrier. Okay? So you can go from one condition to the other, and you'll have a barrier or no barrier present. Okay? And this is something which I think nobody in the field had recognized. And so the lesson really is that you can have a completely rugged energy landscape when you add denaturant, you're smoothing part of it. And the important point is this, as you know, most folding reactions are done by taking protein in high urea guanidine and diluting it out. And people see cooperative reaction. But what this result indicates is the cooperative which manifests itself is purely because of the presence of the denaturant. It's a consequence of the effect of the denaturant on the relative stabilities of the states involved. Okay? It's got nothing to do with what happens in the absence of denaturant. Okay? So if this is true, you should be able to go from here to here by destabilization of N. By destabilization, you're bringing it higher up in energy from here to here. And what we did was show that. So here it is the wild type protein, continuous behavior. You start destabilizing the protein by mutation. We made mutations in different parts which destabilize it. It becomes more and more cooperative. Okay? So so what we found was that 6 to 8 sites exchange cooperatively in all the destabilized mutant variants. And so this was quite, we did other ways of stabilizing, destabilizing, and we saw similar results. So what we took one of the destabilized mutations. There are 6 guys which do it cooperatively, go from one population to the other. We added a little bit of denaturant. Now 14 did it cooperatively. Okay, so instead of 2 fingers coming out together, 4 fingers came out together. Okay? And this is what happens when you add guanidine even to the wild type protein, the one without any mutations. So this vindicated our belief about what's happening. You have 6 sites which are exchange. We are looking at one fragment. So I didn't mention that when you do the mass spec, you can fragment the protein, look at what exchange is happening in every fragment. So you can get information on different parts of the protein. And we could, for example, get information on this sequence segment of the protein. And what's happening there is 6 sites exchange cooperatively. For the mutant, you add guanidine, 14 do it cooperatively. So the denaturant is smoothing out the energy landscape. From going bumpy, it's become smooth.
Jayant, I have a point to make in the previous slide. You can say smoothing. No, going to the slide that you showed with the free energy. Yes. At the end of the day, you're still placing the unfolded state at a higher energy than the native in presence of the denaturant, which I don't agree to, because if you include the free energy of all the constituents of the system now, you're plotting the free energy parts per protein for the protein part.
Well,
but
so
you should pull
okay,
down
yeah,
the
so
unfolded.
it's a little, yeah, I should have drawn the picture.
Right.
The idea behind this was just to show the bumpiness going.
The transition, yes.
It's not completely wrong. It's not actually wrong, because we are doing, the amount of guanidine we added was just 1 molar. And in 1 molar, this fellow is still more stable than this. It's not completely unfolded. So I take back what I said. See, when you, so yeah.
How to view free energy versus potential energy surfaces,
Hmm.
and especially when you include the concentration of the deviation, because that is the system.
No, so we have added just a small bit of small amount, so it doesn't come down fully. It'll come down from here to come down a bit. Okay. So because we could look at different fragments, we could see what's happening at different parts of the protein as it unfolds over 36 days. And this is just showing it what's happening in color. It's a bit misleading because what is red here is all structure which is unfolded, has gone off. Okay. So with time, you have more and more, the greater the exchange means the greater the amount of structure which is lost. I'm showing it this way to just tell you which structure is lost at what time. And you can see even for the helix is not being lost in 1 step. It's going in at least 2 steps. And that really went against what is thought about peptides forming helices, that the helix is there or not there. Okay. This is very clearly not doing it in 1 step. It's doing it in a diffuse manner. And that's true for different parts of the protein. And we wanted to understand why that happens. And again, we used a method called thiol exchange labeling, which is similar to the hydrogen exchange method. But except you can look at what's happening at the tertiary structure level, at the side chain level of protein. All these amide exchange, hydrogen exchange are telling you what's happening at the peptide bond, the main chain of the protein. This tells you what's happening at the side chain. So what you can do is put a cysteine residue at different points. The cysteine is a thiol which will react when it is exposed, when it is unstructured. It will not react when it is buried by structure. So you can tell when a particular residue becomes exposed. And we did that by putting a cysteine here or here or here or here at different points of contact between the helix and the beta sheet, which is behind it. And we could show, for example, that at this intermediate, cysteine 13 becomes exposed in this cysteine 42, in this cysteine 74 and 63. And we could measure the rates also of exposure. It's a tedious experiment to do. It's a very simple no-frills experiment, but it gives a lot of information. Okay, and then we could correlate it with what's happening at the main chain. And so the idea is very simple. I'll just quickly tell you, is that for the helix to unfold, there are some critical interactions at the side chain level. It's only when those side chain interactions are broken that helix can unravel. Okay, so the helix unravels up to this stage when this interaction is broken. For it to unravel even more, this particular interaction has to be broken.
Yeah. Is there any proline isomerization involved here? Is
No.
It's very slow. Without that, even it's slow.
We're looking at without absence of denaturant. The protein is just unfolding by fluctuating. So it will take forever to do it.
Okay.
Yeah, it's spontaneous without all by itself. Proteins are very dynamic, so they're sampling all conformations over time, including the completely unfolded state. Okay, so more lessons. So we go up so what this means is that it's somewhat similar to climbing a staircase. You climb up one flight, then it takes you a little pause before you climb up the second flight, and then there's a little pause, you have to walk around the, and so you have these kinetic pauses. And in the kinetic pauses, you have kinetic pauses here because some specific interactions have to break, and then you can climb further. Again, you have to break another interaction, then you can climb further. That's what's happening.
Pauses, do they follow some law, or I mean?
Well, there's no law to it.
Totally random.
No, so as I said, if this fellow breaks first, then this. Now you might ask, why does this fellow break after this? I don't know, and I don't think one can answer that question. Okay. It's like you're supported out here. If you start fraying out this way, then this fellow will naturally have to break first, and then this. So maybe there's some mechanical effect, or I don't know the answer to that. Yeah.
Can one just imagine it as different directions in a multi-dimensional space of conformations with different activation energies to go different directions? That's probably too simplistic, but just to ask.
It's possible, but I don't. Someone like Ravi might be able to answer that. I have a very. I don't know enough or too much or anything at all about computational methods. So you go up this, and it takes 36 days to unfold completely without any denaturing. Yes?
So my question is, if a protein is going from a folded state to a unfolded state, then it is going through some molten globules, right? If I reverse the process, then will it go through the same molten globules? Like
So
if
that's what I was
refold
just
it?
going to say. This experiment is done under zero denaturant. Any reaction by the law of mass action should happen exactly the same way, backward and forward. Okay, but the problem in protein folding is that you have to fold and unfold under different conditions. So how do you equate? Here I'm doing unfolding and zero denaturant. That's the condition which the protein normally will just fold. Its folded state is stable. So whatever is happening in unfolding has to be the exact reverse of what is happening in folding. So you know exactly what's happening to folding. So if this change is happening early during unfolding, it's happening last during folding. If you were to do folding under normal conditions without any additive. So you can do other things like, again, you can add, this is work with some other protein which is cooperative to start with, but you add something to stabilize the native state, and it becomes non-cooperative. It becomes a continuous gradual chain. So you can play around with this as we have done. And for this protein, which is an SH3 domain from a signaling protein, you can show that there's a possibility of many different ways of unfolding. And what's very interesting here from this result was what's happening during the process is different, because here 4 strands of the beta sheet are doing it cooperatively, whereas here only 3 of them are doing it cooperatively. So this route is different from this in a major manner. Okay? And so this is all from what are essentially, equilibrium, very simple experiments to do. Now, so this brings me to the last part of my talk, the question of how many pathways are there and how do you distinguish between them? And so do you have one pathway for folding and one for unfolding, or do you have many different routes? And about 25 years ago, we did this study doing real-time NMR, where we did the folding unfolding experiment in the NMR tube. TIFR had just got a brand new 600 megahertz machine at that time, and before anything happened to it, we did. I don't think the people in charge knew what we were doing, but we put in plastic tubes right into the magnet and mixed things right in the tube and looked at changes happening with time down there. And we ended up with pictures like this, of our folding funnel and our unfolding upside down funnel. And at this time, I was a bit discouraged because I said, if you have many pathways, how does one distinguish between them? You can't really, from one-dimensional NMR, get any structural information on what's happening. And at the same time, hydrogen exchange and NMR experiments, mainly from Walter Englander's lab, had indicated that you could put the native and partially unfolded states, puffs as we call them, in a ladder such as this, energetic ladder. Okay, so this fellow is the most stable. This fellow, which is partially unfolded in the regions which are shown in light pink, are unfolded there and folded in the colored region. This is more unfolded. This is completely unfolded. And you say that this is more stable, unstable. This is more unstable. This is least stable. And then if you order them in an energetic manner, you can make the mistake of saying that this is also the kinetic sequence of events, which is usually a mistake. Okay. And I'm showing you data not from Englander, but from our lab on the prion protein recently, where we showed that this is not really true. And we could show, for example, that this second unfolded state is actually an ensemble of two subpopulations, and this fellow is not on the pathway to this. And this is very recent work where by doing mutations which affected the stabilities of these species in a differential manner, we could make this claim. Okay, so proteins look like they've been assembled from their parts. So this is your little helix and your beta sheet. Maybe I should have shown this slide right in the beginning to explain protein structure. But you can see that you could probably assemble the whole guy by just taking the parts and putting them together. And is that really what happens? And these ideas are there. So this is the computational method from a paper from Kendall, where again, it's the analogy of the jigsaw puzzle assembly, where you have this protein out here, and it can start folding from any part. And then you can have all these arrows connecting each other. These are all isoenergetic, these are isoenergetic. And so you will see this type of ladder going up. This is what is happening. And again, we resorted to doing FRET, again with G. Krishnamurthy. And we first published a paper with Deepak Dhar again, because again, the kinetics was somewhat complicated and we needed his help and assistance in analyzing the data. And what we could show is that the molecule contracts continuously on two different major routes. And in one route, it gets trapped in some metastable state. And then to go from here to the final native state is an activated process. And to describe the contraction, Sandhya, student with Deepak's help, used a coarse-grained Markov evolution model to describe the process. And because Deepak likes glasses, I don't know what. So in glasses, viscosity is quite high, and so things get trapped. And so typically, about 10 million to higher poise is the viscosity. So Deepak did a back of the envelope calculation for the time scale of the contraction, and we arrived at a viscosity of just within this glassy limit. Okay, and so it looks like this contraction is impeded by whatever impedes glass behavior, which I don't know too much about. But anyway, we decided to use FRET with 4 different distances to look at folding. So this took 12 years after our unfolding paper. And so I won't really describe the data in any detail, except that for one distance just changed in a gradual, continuous manner. The other distance, fraction of molecules with different distances behave very differently. So in all cases, there was a fast phase and a slow phase, but the relative amplitudes were different, indicated that different amounts of molecules were doing it fast for this distance, and for the green distance. So which meant that, if all the distances were behaving the same, you should see the same relative amplitude for the fast and the slow, but you don't see that. And how do we explain that? So this was difficult, but Sandhya finally, just by counting molecules, figured out that you could group them into 4 different categories, one in which all 4 distances were unfolded like, one in which two distances were native like, one in which 2 other distances were native like, and one in which all 3 distances being monitored were native like. And by just counting molecules and enumerating them, we don't know whether that's probably not a unique solution, but it works, and any other solution will give you the same type of result. We could, again, hung. We came up with our folding funnel placed sideways. And what's important is red means things which are happening fast, green is slow, and black is very fast. Okay. So now when I say IB, it means the distance B corresponding to the beta sheet has formed very fast in these 22 molecules. And then some changes happening. The E, the other distance, becomes native like in a fast phase. And then the third C distance becomes native like in a slow phase. And then you arrive at this molten globule someone referred to, and then you go to the native state. So what's important here is to understand, for example, that in this case, the B distance, the distance in the beta sheet, is forming very fast in this 22 molecules. Whereas in this set of molecules, this 22 molecules from here, the B distance is becoming native-like in only the slow phase. Okay, so the same thing is happening very fast here, is happening slow here. Similarly, there are things which are happening fast in one pathway, but happening slow in the other pathway. So the same structural change can happen fast on this route, whereas it occurs slow on the other route. But the actual rate at which it happens is not different across the different parts of the protein you are looking at. And so this is really something which, we had been trying to figure out for a long time, how do you structurally differentiate multiple pathways of folding. And the other thing which I just want to mention very quickly, because I've just shot past, is that we worked with the 2-chain version of this protein. We did the hydrogen exchange experiments. We could show that what's happening at different parts of the molecule in 2 different folding conditions, and very low denaturant and slightly higher denaturant, where we had shown in an earlier study that the pathways are different. And what you can see, for example, here is there's no green in this panel and this panel, but there is also no green here, but there is green out here. What the green is telling you is this segment is forming protective structure and then losing it in an intermediate and then losing it. And that's happening for all the parts where you're seeing green. But here there is no intermediate, there's no protective structure in the intermediate for this segment and for this segment. Whereas when you do it at a higher denaturant, the intermediate does have protective structure in beta 2. Here it doesn't have protective structure in beta 2. So the pathways are different in the 2 folding conditions. And that's again something which in the field has been very difficult to show for any particular protein. So the pathways, so you can see this has got two blues out here. Here, there's only one blue out here. Otherwise, the pathways are quite similar, except the extent of structure in the intermediate is different because you can see the coloring is different. So, so the final message I want to tell you from the last part of the talk is that there are multiple pathways. You can characterize these multiple pathways. Some things happen fast on one pathway, the same thing happens slow on another pathway. And these results really contradict that fold-down model which I referred to, which says that folding must occur in a unique manner on a single defined pathway. So with that, I'll conclude. The work I talked about from my lab was done by these two students primarily, and the student currently still in the lab. I've benefited a lot from my collaboration with Krishnamurti over more than 25 years, and it was indeed a great privilege for me to have worked with him. I've also collaborated with many other faculty members at TIFR. I probably collaborated with more TIFR people than anyone else. I should mention my papers with Deepak are probably the only papers still from TIFR which has got a physicist, chemist, and a biologist together. It's a shame, but there should be more. Okay, and of course the 30-odd students, PhD students, and MSc students and postdocs whose collaboration has been, like, invaluable. So thank you for your attention.
Thank you, Jayant, for a very nice, sort of rendition of all the things that you have done and sort of captured the last part, which is the heterogeneity in the pathways.
So I talked about the bold students' work. I didn't talk about the other.
Oh, well, there are lots of very interesting information in the folding part. So the colloquium is open for questions. Yes, further questions.
Hi, Jayant. Very nice work carried out over many decades. I think almost all of your papers are really landmark papers in the field.
Oh.
So I like especially this work, recent work, where actually have done hydrogen deuterium exchange over 36 days. That's one of the most beautiful experiment. But I'm trying to understand, so let's say when the protein completely deuterated, okay? So that's not really denatured state. That's still folded state.
No. So it takes a certain amount of time to sample
Different.
the completely unfolded state, but then it comes back. So what this method tells you is how long has it taken to sample the completely unfolded state.
For the entire protein structure to expose to
for
this.
every part of the protein structure to sample the completely unfolded state.
Correct, because
so that's the time of unfolding.
It's correct, but the landscape picture shows that somehow I think it's not correct entirely, because in the end it's not really denatured state. It's still, you can have completely deuterated protein but folded state.
No,
right, but
if you're doing the experiment under native conditions, you end up always with native protein.
Correct.
It's just that the method you're using is telling you that this protein has sampled all sorts of states, sampled this. It takes this much time to sample the completely unfolded state.
Yeah.
And then it zips back right away.
It folds
But
back.
We are not bothered about that because we are just looking at how long it's taken to do this.
Okay. Because the picture somehow, I think, people might misunderstand that in the end, the protein might be really denatured after 36 days.
I should have
36 days. Okay. That is beautiful experiment.
Okay, I have two questions. One is, so in case of chemical unfolding and refolding, the time scale at which it refolds, do you think it will follow the same when a new in the living cell, when protein synthesizing and then coming out and then doing the same thing? Or say, for example, when protein in unfolded conformation is passing through a channel and then folding, say inside the ER, do you think the similar kind of time scale it will follow?
offline cells also. So except you need to think about the environment itself. So the cell is a very crowded environment. So we know how crowding affects chemical reaction. Okay, so you know that what is going to happen purely because of viscosity effects or crowding effects, etc. But there can be in the cell specific molecules which interact, which might modulate the process. So the role of in vitro work, which is what we do, is to really lay out the possibilities.
Yeah, that's good.
Okay.
Yeah.
It's possible, sorry, for it to happen in this way or this way or this way. And the cell, whether it's happy or sick or whatever, will do it one way or the other.
Yeah. I was the second question of mine was in this refolding experiment, you have the entire sequence available where it is kind of doing this, right?
Mm.
But when it is doing in the cell, it is actually a sequential way it is coming out, right?
Yeah.
It is not like it is coming the N terminal first
Yeah.
and then coming the C terminal
Yeah.
and that way. So that I was just wondering how we can.
Yeah, so there's quite a bit of data now which says that you really need the full chain to come out from the ribosome before native structure can start folding. Okay? And obviously, when the chain is poking out and coming more and more, protruding more and more out from the ribosome, it will form some non-native structures.
Yeah.
But those are non-native and they finally have to.
Shudipta?
Yeah, a quick question which follows Koti's, that you are assuming that whenever there is a hydrogen exchange, it must have opened up, even if it's once in 36 days.
Yeah.
What about small fluctuations which are already there, always there, without fully opening up? It is just a slow exchange. Somehow the deuterium finds its way because of small fluctuations. It never really opened up, but it got exchanged. It's just slower. How do you differentiate between the two?
So it's difficult. So, and there has been, there's no real way of differentiating between that, except that there's practically been no report of water entering into the core of a protein from the outside. So for exchange to happen, water has to enter right into the hydrophobic interior.
But small fluctuations which are there at KT,
Mm.
a priori, I don't see if it's impossible. It won't be there in
So
the
it's
protein
not
structure
impossible.
because it will be a very rare
It's not
event.
impossible, and
So 36 days is also a long time. So it has taken a long time. So it's a
Mm.
very unlikely event. So you'd never see it in a crystal structure or something.
Mm.
Just posing the question.
No, I don't have a proper answer to that. That it's possible that there's some little water channels which form because of localized fluctuations, and
So that's a long time.
So, well, thank you, Jayant, for such a wonderful talk. And I think also not just the quality, but the interdisciplinarity is a example and inspiration to us all. So thank you. I've got a very naive question again, which is to go back to the start of your talk and AI and DeepMind and so on. Let's say we now have the structures for several proteins, but we want to figure out the dynamics for how they unfold and so on. Can we put that structure to use in the following very naive way? Is it possible to combine the knowledge of the structure with, say, the in vitro experiments to find out what might be the rate limiting steps? So maybe there are many paths, but we can figure out just in terms of conformations or something, what the rate limiting steps are. And they may, in the end, be the most important ones for the timescales involved. So we don't have a fine-grained view of exact trajectories, but a coarser-grained view of the choke points, and from there, an estimate of timescales. Is something like that?
I don't know enough about these methods. Maybe you can, but the computer will get to know what's happening, but I don't know whether humans will learn what the computer knows.
The question is about using the experiment. Like you know
Yeah, but,
From your exchange experiments, which portions are
yeah,
more?
yeah, but if you want to use it as a learning basis, you need many such experiments
That's to be done
done.
on multiple structures.
Yeah.
Similar.
Yeah, so similar kinds of structures, you need a group of experiments, you need,
And then you might be. able-
AlphaFold works because it's got 500,000 structures to train itself on. Or more than 500,000 now, I don't know what the number is. No, it's made 200 million structures, but it got trained on 500,000. Yeah, so that's 500,000. No, but I think that's going to be the future, that you should be able to do it once there's enough data to train these programs on. And then many of us can retire.
No, I didn't mean that. I meant we.
No,
combine-
no,
Yeah,
I mean, I think if we can get an understanding of why it's happening in a particular way, it would be great. So right now, for example, there's very little understanding. AlphaFold is a completely probably what you'd call a statistical method. There's no real physics in it. If physics gets incorporated, that might help us understand how it is predicting structures. But
Just a quick thing essentially. So if you could potentially use physics-based models to unfold a protein using known forces, etc. And if we have his experimental data in terms of all the different ensembles, one could validate those. And then using this plethora of structures that AlphaFold has generated, you could repeat it over and over again on the computer and potentially derive that information that you're thinking about. So in that way, the experiments provide you with the kind of validation point for the physics-based models to look at the intermediates, which AlphaFold doesn't have. Right. Yeah. Right. Right. Yeah.
But I think one of the major concerns would be, Jayant would agree, the experiments have to be uniformly done for different kinds
Yeah.
of classes of protein. That is something that was a challenge from different labs and different situations and conditions. For whatever.
well, the training base for
Training base.
structures is not done. All the crystals are not formed under the same condition.
Right.
But it still works.
Is it time for one question along the same? Like see your final picture that emerged, right, in terms of all the different intermediates, right? That was all derived from essentially experiments like the FRET experiments, for example, the thiol exchange, etc. But these are projections on the free energy landscape, right?
Yeah.
Like, so I was just wondering, essentially, how did you guys actually put those projections all together to form this? Was there a model involved in that, or is there any, was it?
No, so
How unique is it?
there's no model. I mean, there are kinetic models in terms of, where you fit in your variables to a kinetic model. Now, that's about it. So in terms of projecting it on one particular landscape, I mean, one particular reaction coordinate, the reaction coordinate is the experimental measure. Okay, so distance between points, or the extent of exchange which has happened. Now, how do you go from this experimental reaction coordinate to a coordinate such as number of native contacts? I don't know how to do it. I don't think there's any simple way, because number of native contacts also is, which all computational people use, that they can have many isoenergetic conformations which have the same number of native contacts.
Yeah, I would like to add, because you brought the Marcus picture. Marcus has always said, for any chemical reaction, there are only 2 coordinates that matter. So it appears the problem is huge, but it could be just few.
So I don't know on what basis Marcus said that. So
The reaction
It-
coordinate by definition should be.
it doesn't necessarily have to be true.
Right. by this, yes, happen. Okay, well,
Okay.
on that note, let's actually thank Jayant for
Thank you.
putting a profound talk, and I'll thank all the audience for participating in the discussion. Let's get some food for the hungry stomachs. So that's outside the canteen.